Hello viewers, welcome back to Trendy Achievers channel. I'm back with one more important concept in zoology, IPE second year, that is immunology concept. What is immunology? Before knowing about immunology, let's know about immunity. The power of fighting against the foreign object that enter into the body called as immunity. So then అంటే ఏంటి ఏదైనా ఫారెన్ ఆబ్జెక్ట్ బాడీలోకి ఎంటర్ అయినప్పుడు ఫైట్ చేసే కెపాసిటీనే ఇమ్యూనాలజీ. Immunity. Then what is immunology? Immunology is the combination of different organs, cells and their products and some of the proteins which are involved in the immunity. The study of the total immunity is called as immunology. That means what finally? Immunology is the branch of biology that deals with the immunity and to kill the objects, foreign objects that enter into the body with the help of different organs, cells, proteins like antibodies and different cells that involved called as immunology. So we got an idea what is immunology. Then what is here today? We are going to observe the important proteins which involves in the fighting with the organisms or foreign objects called as antibodies or immunoglobulins. They are the important proteins which are released by the B lymphocytes. B lymphocytes are what? They are the cells, they participate, they play a very key role in the production of antibodies in our body. So now let's know today about the structure of immunoglobulin. Very important concept for IPE as well as in the NEET. So students, don't stop the video till end, okay? If you watch that, you get a clear idea about this. So now this is the structure of an immunoglobulin or antibody structure. It looks like a Y structure. So when a foreign object enters into the body, our B lymphocytes releases a group of proteins called as antibodies. So that antibodies are produced by the B lymphocytes, and in this is the structure of immunoglobulin or antibody structure. It consists of two types of chains called as heavy chains and light chains. So that's why we call it as a H2L2. H, heavy chains 2, 1 and 2, light chains 2, 1 and 2. Okay students, now let's see. So this is the region where the antigen, this is the site where the antigens ekkada recognize cheyabadtayi, ikkada. A site where the antigens are fixing with the antibody called as antigen binding site or fragment antigen binding. So this is the fragment where the antigen fixes here. That area we call it as a paratop. Very important term. So paratop. So we can observe that this is the variable regions and they are the constant regions. And at the end, fragment crystallization end we can observe. And here these two fragments, these two chains links with the help of sulfide bonds. And here the immunoglobulin structure was first observed by Rodney Porter, and based on their location, the immunoglobulins are of two types, free and then surface. If we observe the free, they used to present in the circulation of body fluids, and the surface they present on the B lymphocytes and memory cells, they present as a surface antibodies. Okay, so based on their monomeric or pentameric, they classified into 5 types. They are IgD, IgE, IgG, IgA and IgM. So very important for NEET students. So here these first 3, IgD, IgE and IgG are monomeric, and this is the IgM is the pentameric and IgA is the dimeric. So this is the structure of immunoglobulin, students. I'll come back with one more immunology concept. I think that this is a very useful for you in the case of IPE as well as in the NEET. Thank you.